Today I get the honor to introduce Dr. Kathy Green. She is a professor of dermatology and the associate director in the basic science division at the R.H. Lurie Comprehensive Cancer Center at Northwestern University. For starters, I'll say she is an extremely prominent researcher within the intermediate community. So it made it really difficult for me to make a very confined introduction. But she's done really wonderful work. In 2023, she was awarded the Dry Skill Graduate Program Faculty Teaching Award. She has a total of 208 publications, and 148 of those are peer-reviewed articles. These articles, I'm sure many of us have read, but they focus on desmosomes in cancer, dependence of gap junctions on desmosomes, as well as desmosomes and intermediate filament connections, and mechanical forces. With her conducting this work, she is not alone. She has trained over 20 PhD students and over 30 postdocs, many of which have obtained different levels of staying in academic research. In addition to her research, Dr. Green also serves as a treasurer for both the International Societies for Investigative Dermatology and for the Society for Investigative Dermatology. Sorry, I think I read that twice. She serves as a deputy and associate editor for multiple journals. So with all that being said, I am very excited to hear your work, and I hope everybody gets to learn something new today. So thank you, Dr. Green, for presenting, and we look forward to hearing from you.
Well, thank you very much, Clara, and also your co-organizers, Lonnie, Nuria, Elena, and Emma. I really thank all of you for inviting me. This is really a great opportunity to connect with everyone all over the world, and I really appreciate the invitation. So I'm going to start sharing my screen now. Okay, Clara, do we look good here?
Yep, you're in presentation mode.
Okay, perfect. So as Clara said, we're interested in our laboratory in desmosomes and desmosome intermediate filament connections. And desmosomes have arisen not only as mediators of cell-cell adhesion, which is absolutely critical for tissue integrity, but they're also signaling nodes. And a lot of us in the field, and I know there's people on this call like McDowell Hatzfeld and others, who are very interested in these non-canonical roles for desmosomes. So today we're going to be talking a little bit about those. Now, my story really begins many, many, many years ago, thousands and thousands of years ago, in single-celled organisms called choanoflagellates. And in choanoflagellates, we see for the first time primitive cadherins, and these are calcium-dependent cell-cell adhesion molecules. You might ask, well, what are these cadherins doing in single-celled organisms? Well, it was thought they might have been involved in capturing prey. And then later on, those cadherins were co-opted by multicellular wannabes like ancient sponges, which brought those cells together into multicellular sheets and also provided the opportunity for organizing the cortical actin cytoskeleton. So this is where we see primitive adherens junctions, and these are the precursors to desmosomes, which we see arise during evolution in vertebrates. So fast forward to vertebrates, we see the appearance of desmosomes and their associated intermediate filament connections at a time when organisms were getting larger and the tissues were getting more complex. And then when we fast forward again to terrestrial vertebrates coming out, living life on land, experiencing a lot of environmental challenges, larger organs, we see the expansion of the desmosomal cadherins and also an expansion of their associated keratin intermediate filament proteins. And it's our contention that creating this new sort of more recent intermediate filament desmosome connection allowed us to develop better barriers to live life on land. Now, in this slide, we introduce the importance of desmosomes in mechanical integrity. And you can see over here on the left in these videos that if we introduce a desmoplakin uncoupling mutant, so this is a protein, desmoplakin, that's an essential anchor for intermediate filaments at desmosomes. And if we introduce a mutation into epithelial keratinocytes that uncouples intermediate filaments from desmosomes, you can see that these epithelial sheets fall apart very easily. This is critically important, not only for epithelia, but also for other tissues, such as cardiac tissues, where desmosomes are part of the intercalated disc. And an intermixed type of junction in mammals called the area composita. And mutations in desmosome molecules lead to a number of different disorders of the epithelia, including the epidermis, which is going to be the model system I'm focusing on most today, but also different types of cardiac disorders like arrhythmogenic cardiomyopathy, for instance. And actually, there's been a distinct form of cardiac disorder, myocarditis, that is caused by mutations in desmoplakin. And these disorders are not only disorders of fragility, but they're also disorders of inflammation. And today I'm going to try to connect the physical properties conferred by desmosomes with the inflammatory properties that occur in their absence. So the story today will be about how desmosomes create the physical barrier, or help create the physical barrier using epidermis as a model system, but also how they play a role in developing the immune system. And I'm going to try to convince you, in fact, that there is a molecular complex, and it's actually an actin remodeling complex, interestingly, that connects these two functions of desmosomes, creating the physical barrier and then creating the immune barrier. And we're going to start with a story about how desmosomes help create the physical barrier. And again, as I mentioned, we're going to use epidermis as a model system. Of course, the epidermis is the tissue, the outer layer of our body that is the first defense against the environment. And it has multiple functional layers. It provides a physical barrier, protects from water loss. It also has ultraviolet radiation protection features and an immune barrier. Now, how does the epidermis, how does it regenerate? How does it create it? Well, it regenerates through about every 28 days through a process of proliferation of cells in the basal layer where the stem cells reside, and then a commitment, a cell-fate commitment. Those cells move into the superficial layers. Eventually, they exit the cell cycle. They undergo a well-orchestrated process of differentiation, which is a biochemical differentiation process, but also the elaboration of new proteins, new lipids, and eventually the formation of the life-preserving cornified envelope. And desmosomes play a critical role in every step of this regenerative process. And today we're going to be talking about the mechanisms by which this happens at the cellular level, and showing this really cool video from Corinne Neeson's laboratory, where we're looking at the multi-layering process through both cell divisions, but also the process of delamination, which is shown here. And that is single cells that are on the basement membrane they dissociate from the basement membrane, and then they move into the superficial layers. And we'll be talking a lot about this delamination process today, and then how this multi-layering actually leads also to the formation of the immune barrier. So now I want to just take a page out of our cell biology textbooks and just make sure we're on the same page about intercellular junctions and how desmosomes fits into this whole scheme of intercellular junctions. So on the left, we're showing simple epithelial cells. Intercellular junctions in simple epithelia are polarized along the lateral membrane, starting with the tight junctions up here at the top at the apical region. Tight junctions, of course, provide a very important barrier for simple and stratified epithelia. They keep proteins where they're supposed to be, and they control passage of different components from the lumen down through into the bloodstream. And then we have our adherens junctions, which I already introduced to you as being the ancient cadherin-based junctions that associate with actin. And then we have our desmosomes, which are part of the junctional complex, but they are also along the whole lateral border of simple epithelia. Now, the blueprint for adherens junctions and desmosomes shown over here on the right is very similar. So in both cases, you have transmembrane cadherins. In the case of adherens junctions, the classic adherens like E-cadherin. And in the case of desmosomes, we have desmosomal cadherins. And these can be divided into two classes, desmoglians and desmocollins. And these proteins work together in ways we're still trying to understand to mediate adhesion in the desmosome. And then we have our armadillo proteins like beta-catenin and alpha and P120-catenin and adherens junctions, and plakoglobin and plakophilins in desmosomes. And those associate with the cytoskeletal linking proteins alpha-catenin and desmoplakin, respectively. Now, in addition to having a similar blueprint, these two types of junctions also work very closely together functionally. In fact, you can't really dissociate their functions because desmosomes and their intermediate filament connections are really important for balancing the contractile activity generated at adherens junctions within an epithelium. And one of the things that I'm going to be talking about today is how desmosomes can actually change the localization of something called vinculin. Vinculin is a surrogate indicator of tension on adherens junctions, and desmosomes actually have a pretty powerful impact on whether or not vinculin will be present at adherens junctions. So that's something that will pop up later in the talk. Okay, now that's the simple situation and simple epithelia, even though it's not so simple, but what happens when we go to a multi-layered stratified tissue like the epidermis? Well, in this case, intercellular junctions are also polarized. In particular, desmosomes are polarized in large part due to the fact that there are 7 different desmosomal cadherin genes giving rise to at least 10 isoforms. And these are present at in a differentiation-dependent pattern from the basal to the apical multi-layered tissue. So some desmoglians, for instance, are on the basal layers, proliferating layers, and others are more concentrated in the superficial layers. And it's the idea in my laboratory that overlaying this polarized desmosomal cadherin intermediate filament system on the more ancient classic adherin actin system provides tissues with new opportunities to develop structural and functional complexity. And one of the heroes of today's story is a desmosomal cadherin called desmoglian 1. And we're really interested in this desmoglian because it actually appeared very late in evolution in terrestrial or previously terrestrial organisms when we came out onto land. It's only present in stratified tissues, and it's first expressed in cells in the basal layer that are beginning to commit to differentiate and move into the superficial layers through the process of stratification. So we're wondering whether or not it may actually play a role in the process of stratification. So we're interested in general in desmoglian 1 and its associated intermediate filament system in development of the multi-layered epidermis. And toward this end, we're actually looking during epidermal development in animals, and we also use in vitro models that I'll be presenting to you to look during this process of the development or the multi-layering of an epithelium. Now, this process comprises a number of different mechanisms like asymmetric divisions, whereby cells in the basal layer change the position, the orientation of their mitotic spindle from parallel to the basement membrane to orthogonal. But it also, as I introduced previously, involves a process of delamination whereby single cells move from the basement membrane into superficial layers. And during this process of embryonic development, desmoglian 1, this desmosomal cadherin, actually comes on around E14 at a time when the stratification process is really ramping up. So to address whether or not desmoglian 1 may be playing a role in this process, we made a knockout animal model. This team over here, led by Lisa Godsell, knocked out all 3. There are 3 desmoglian genes in mice, unlike humans, which have 1. We knocked them all out through CRISPR-Cas. And what we got actually was a perinatal lethal phenotype. The superficial layers actually all peel off, sort of like the disorder called pemphigus. And this is actually something that was also reported by Jan Swaschke. But what we've been doing now is looking during development to see whether or not the stratification process is impaired. And toward that end, Lisa measured the thickness of the epidermis early on when desmoglian 1 is just coming on, E14.5 and 15.5, and saw, in fact, that that's significantly thinner. So early on, loss of desmoglian 1 leads to a thinning of the epidermis, but then later on, the epidermis catches up. And we think this is through compensatory mechanisms, and we're looking very carefully at what those mechanisms might be. So to begin to address what desmoglian 1 is doing at a transcriptional level during these early thinner stages and then the later compensatory stages, we're doing single cell RNA-seq. And this is a work in progress. And we're looking at all stages all during development. And here I'm showing you the keratinocyte clustering that we've done to show that there are a couple of different basal populations, as well as a transitional population that expresses both the basal keratins and the superficial keratins, and then the differentiated population. And in particular, we've been interested, this team here is looking very closely, and I'd like to shout out Bree Jarrell and Ziyu Ren, who are currently working this up. And we're very interested in two pathways that are emerging. One of this is alterations in transcriptional pathways controlling actin genes. And in particular, I've highlighted vinculin here. Remember, I told you that vinculin is an indicator of tension on adherens junctions, and it's significantly downregulated in the desmoglein knockout animals, which we find very interesting. But we also see alterations in cell cycle signatures, and we see that there are increased cycling cells in the superficial layers or pathways indicating this, signatures indicating this. And we can actually do staining to show that there's an increase in Ki67 and other indicators of cell cycle. And so we're interested in the coordination of actin or cytoskeletal programs and cell cycle programs, and in particular, an inhibitor of this phospho-RB-E2F pathway called BTG2, which is downregulated. And we're looking at the possibility that this may be involved in keeping cells in the cell cycle later on so that they could perhaps catch up and give rise to a full thickness epidermis by E18.5. So this is a work in progress. We're validating these now, but it leads to a model whereby we propose desmoglein may be integrating proliferation and cytoskeletal programs during epidermal development to lead to the multi-layered epidermis. But this is all transcriptional pathways in the process of being validated. Can we look mechanistically at the role of desmoglein 1 in this process? And toward that end, we have a couple of in vitro models. This one is a human reconstructed 3D model of epidermal development, whereby we take cells from human tissues, put them into submerged culture on top of a fibroblast feeder layer, then lift them to an air-medium interface, and this triggers the multi-layering and differentiation formation of something that over here on the right you can see looks remarkably like human epidermis. And we can manipulate these cultures by introducing retroviruses that have shRNAs or proteins and their mutant variants to allow us to study their role in the formation of these multi-layered cultures. And a number of years ago now, so this is some very foundational work that was carried out by Spiro Getzios and Corey Simpson in the lab in the 2000s, where they used this model system, knocked down desmoglein 1 to address what it does during the multi-layering of, during this epidermal development. And what they saw was something, and I'm not showing you these data, but they saw that MAP kinase signaling, EGF receptor dependent and ERK signaling was elevated in these cultures. So they concluded that desmoglein 1 is normally necessary to suppress, to keep control over MAP kinase signaling in order to allow differentiation to proceed. But shown here, they also saw something very interesting about the morphology of these cultures. They saw evidence of a loss of differentiation characteristics, like the keratohyalin granules are gone. They saw changes in cell shape, shown here outlined in actin staining. They saw in the control cultures, we saw a very well-orchestrated program of stratification. And occasionally we could see these delaminating or umbrella-shaped cells. But over on the right in the desmoglein deficient cultures, there was a jumble of cells. It appeared that the stratification process was not occurring normally. And in a possible compensatory mechanisms, we saw an enlargement of the superficial cells. So we proposed that loss of desmoglein 1 might lead to an aberrant stratification process. And we wanted to carry out gain and loss of function experiments to address whether this was the case. And when Oksana Nekrasova, a postdoc in my lab, was there, she carried out a very brave experiment. A number of you have seen this data before, but I show it every time because it is so striking. Oksana hypothesized that since desmoglein 1 is only present in stratified tissues, perhaps if you introduce it into simple epithelial cells, you could get them to stratify. So she introduced desmoglein 1 GFP fusion protein into simple canine kidney cells and saw something really interesting. She saw these cells pop up from the monolayer in a process of extrusion or delamination. And instead of undergoing apoptosis, which is what you might have expected to occur, they actually stuck to the top of the monolayer and they formed another layer. So desmoglein-1, when overlaid onto the existing actin and other cytoskeletal networks that are present in these cells, is sufficient to drive stratification of these simple epithelial cells. So how does it do that? How does desmoglein support delamination? Well, in taking a page out of those that study simple epithelial like Jody Rosenblatt, and then people like Sarah Vikstrom who've looked at delamination in stratified tissues, we know that these things over here on the left, breaking mechanical symmetry across the plane of the epithelium, redistribution of cortical tension or the actin cytoskeleton, which is something also shown by Alpha Yap's lab, and also compressive forces in the basal layer are important for this process. So we hypothesize that desmoglein-1 is contributing to these mechanisms. And toward that end, when Josh Broussard was in the laboratory, he decided to take a close look at the actin cytoskeleton because this is what would need to change in order to break mechanical symmetry. And we were inspired by some of these older experiments where we looked in desmoglein containing in cells that are absent desmoglein here in these small differentiating cultures over here on the left. And we can see when we look just at cortical actin, presence of desmoglein-1 is able to reorganize actin and concentrate it near the junctions in the cell cortex. But in the absence of desmoglein-1, you don't have as much cortical actin. You have a lot of actin, but it's all down on the substrate. So Josh decided to carry out some experiments, and this first experiment I'm going to show you is an actin polymerization experiment where we put in fluorescently labeled G-actin, and we look for the polymerization of new actin. And this is through a machine or a protein machinery that contains ARP23 and NWASP. So these are proteins that are important for the polymerization of branched actin. And so here we see what happens when Josh introduced desmoglein-1 into keratinocytes. We could see that this stimulated the polymerization of new actin, shown here by these little red dots around desmosomes. Now, what was the consequence of this polymerization of actin in response to desmoglein expression? Well, we decided to look at the surrogate indicator of tension on adherens junctions called vinculin. And we can see in the control cells, there's plenty of vinculin on adherens junctions, but in the desmoglein expressing cells, there's not as much vinculin, suggesting that the adherens junctions are under less stress. We also confirm this with an e-coherent FRET sensor. Then he wanted to look at more directly at membrane tension, and for that, he carried out laser ablation. And what we can see here is when you ablate the membrane in a control cell, there is a very robust snapback indicating they're under tension. But in the desmoglein expressing cells, there's less snapback, and he can quantify this and show that the desmoglein expressing cells are under less tension. All of this suggested to us then that the expression of desmoglein-1 is able not only to promote differentiation, but also to remodel actin through a process of polymerization and to decrease the tension on adherens junctions. So next we wanted to know how is desmoglein-1, which we normally think of being anchoring intermediate filaments through desmoplaque, and how's it coupled to actin? Well, for that, Mira Hanagazi did a deep dive into the sequence in the tail of desmoglein-1 shown here, and Bree Gerald is following up and carrying on with this project. And what they found is a Nick SH2 domain binding motif right here with a very critical tyrosine residue. Now, Nick and Crick were actually proteins that were found by Terry Leckler through bioID experiments to be in proximity to desmoplaque, and here we see a binding site on desmoglein-1. And so we hypothesized that maybe NIC1 could link desmoglein to the cytoskeleton. It's actually also known that NIC1 is important for activating the NWASP ARP23 actin polymerization complex. So to address if desmoglein binds to NIC1, Jen Kutsier did a GFP trap, and we can see, in fact, that it does bind to NIC1. But if we mutate this critical tyrosine, we can see that abrogates association of NIC1 to desmoglein-1. So what is the importance of this association? So in order to address whether NIC1 might be necessary to redistribute vinculin away from the adherens junctions, we compared its expression to that of wild-type desmogliin. And as I showed you before, wild-type desmogliin can decrease vinculin on adherence junctions, but the Y1042F mutant is unable to do that. So there's still tension on the adherence junctions. Now, what's the consequence on that with respect to the actual stratification process? Well, to address that, we carry out a sorting assay whereby we mix unlabeled cells with labeled cells that are also genetically modified with various mutations, and we mix those, and then we quantify the number of cells, the deviation from the predicted 50-50 distribution of labeled and unlabeled cells in the population. So now, if we introduce desmogliin into cells that don't yet express desmogliin, we can see that the number of stratifying cells goes up. However, if we introduce the Y1042F mutant, this actually not only can you not see that stratification, it almost appears like it's acting like a dominant negative, and you see a lower number of cells that are stratifying in this sorting assay. So that suggests to us then that desmogliin 1 is able to recruit and organize this actin remodeling complex next to desmosomes in order to alter the tension on adherence junctions. We can also see over here the results of additional sorting assays that if you knock down NWASP or NIC1, you can see that you prevent this cell sorting into the superficial layer. Okay, so I told you that desmogliin 1 helps recruit this actin remodeling complex, but I haven't told you whether or not, or addressed whether or not, the association with intermediate filaments is required. Now, of course, this is the canonical function of desmosomes, so it makes sense that we want to know whether or not it is important for supporting the stratification process. So in order to address that, we carried out another sorting assay, and in this case, we introduced desmogliin 1 to promote stratification. And in another arm of the experiment, we knocked down desmoplakin, or we introduced an uncoupling mutant, something that uncouples desmoplakin from intermediate filaments. And what we saw is that that actually prevented desmogliin 1 from promoting stratification. So not only is desmogliin 1 promote stratification, but the intermediate filament connection is necessary for that process. But what's the mechanism? What is the mechanism by which desmoplakin and the association with intermediate filaments promoting stratification? Well, going back to our list of things that are important for delamination to occur, here at the bottom is compression. So it's known that compressive forces are able to squeeze from neighboring cells, are able to squeeze the delaminating cell into the superficial layer. So when Josh Broussard was in the lab, he actually looked in these 3D cultures at the different mechanics in all the different layers. And what he found very interestingly is that at the top, and I'm not showing you this, but those cells are under high tension. And actually, the fact that there's a tension gradient in the epidermis was reported by the Fuchs laboratory, and we also showed it using this assay here that Josh did. And in this little video over here, we're looking at what happens when you laser ablate the basal layer in these maturing cultures. And we see that as they mature, those cells are under compression. So is it possible that the intermediate filament connections are playing a role in this? And so to address that, he carried out these laser ablation assays in control and also cultures expressing this uncoupling mutant over here, DPNTP. And what he could see is that the negative recoil indicating compression is actually impaired in the DPNTP mutant expressing cells. So these cells are under compression in the layer that it's delaminating, but if you uncouple intermediate filaments, that compression doesn't happen. So this leads to a model whereby we suggest that desmogliin 1 recruits actin remodeling machinery to redistribute tension, but that you actually need the intermediate filament connection to confer the compressive forces on these cells that are important for delamination. So this is our current working mechanistic model that we're continuing to test. So now how can we want to go back in vivo? So I showed you an in vivo model of desmogliin deficiency, but now we also want to develop an in vivo model of desmoplakin intermediate filament connection deficiency. And toward that end, we turned our attention to Carvajal disease. And we're particularly interested in this family right here that has a mutation in the C-terminus of desmoplakin that removes the final C-terminal plakin repeat domain C, which is part of the intermediate filament binding domain. And it also removes this sequence of 68 residues, which is a really important unstructured region. And this region contains targets for a number of different post-translational modifications, both by arginine methylation and serine phosphorylation, that we have shown over the years to be a really powerful regulator of the strength of binding of desmoplakin intermediate filaments, and it allows the ability to control adhesive strength of desmosomes. And one of the reasons we're really interested in this family, human family, is that there is a spontaneous mouse mutant called the desmoplakin ruffled mouse, and it has a mutation that's really comparable, almost the same mutation that the humans have. And so we're beginning to bring this mouse, develop this mouse model in the lab to address several questions. Number one, what is the impact on early development and this delamination process that we're interested in? And to that end, we're actually mating these animals with desmoglein 1 fluorescent reporter animals that report cells that are first expressing desmoglein 1 and beginning to adopt this cell fate that will allow them to stratify. And we're also interested in using the Carvajal animals to look for what happens to the mechanics and the polarity of the epidermis in these mice that have impaired intermediate filament binding. And to begin to characterize this model, Rose D. Dominicus in the lab is doing some really beautiful pole mounts, and you can see here on the right, going down through the different layers of the control and the Carvajal animals, that there are changes in the organization of desmoplakin-containing structures, desmosomes, and cell shapes. And over on the left, you can see here more clearly that the desmoplakin structures quite beautifully and are altered in their distribution and their morphology. And we actually think that there are changes in the way the desmosomes assemble in these animals, and we're considering the possibility that there are alterations in phase separation and condensate formation, which we think is part of the mechanism of desmosome assembly. But we're also interested in alterations in cell shape and nuclear positioning. And in collaboration with Mariana in the Center for Advanced Imaging, we're looking at all these different parameters. And very notably, there is a shift in the nuclei within these cells so that they are organized in an off-center manner. And this could have an important impact on chromatin organization and gene transcription and other nuclear functions that we're very interested in. And finally, we're also interested in whether or not this tension gradient that I already mentioned to you, high tension in the superficial layers and lower tension in the basal layers, may be altered due to an alteration in the balance of the contractile actin network and the desmosome intermediate filament network. So Abby Pearl has raised the possibility that this tension gradient may be altered in the Carvajal animals. So not much data yet, but this is a work in progress, and we're really excited about using these animals for intravital imaging and also analysis of some of the other characteristics, including nuclear functions. So what I just told you then was desmoglein 1 and its associated intermediate filament system in desmosomes during epidermal development is important for creating the physical barrier. And now I'm going to end the talk by talking about how desmosomes are important for establishing the immune barrier. Now, in fact, desmoglein 1, even though it comes out in the basal layers, is expressed at its highest levels in the superficial layers where we are exposed to pathogens, ultraviolet radiation, and other irritants. And in fact, we think that it's playing a role as a sensor of all of these environmental stresses. We got interested in that a number of years ago when Jody Johnson was in the laboratory, and she noticed that ultraviolet radiation leads to the temporary reduction of desmoglein 1, and loss of desmoglein 1 in keratinocytes actually elicits the production of cytokines, which are involved in this so-called tanning response. And we were able to take conditioned media from these desmoglein deficient cells, put them onto melanocytes, and amazingly, they produced more melanin. But this was a temporary protective response. And we now know that chronic loss of desmoglein, either in genetic disorders or in the context of melanoma. And we have a whole nother project on melanoma. I'd like to shout out Jenny Picorni and Xin Tong, who work on this area, and I don't have time to talk about it today. But in both of these cases, chronic loss of desmogliin is pathogenic. In order to look more closely at what type of pathways are affected through chronic loss of desmogliin 1, we carried out RNA-seq and saw not only were there changes in keratinization and differentiation programs, but there was a very striking change in pathways involved in antimicrobial response, inflammatory response, wound healing, and defense response to bacterium. So in animals, even before they were born at E18.5, and also human patients with a disorder called SAM syndrome, who are desmogliin deficient, they exhibit an inflammatory signature. Now, what kind of signature is this? Well, working with these folks at University of Michigan, we demonstrated that the signature is similar to that with patients who have psoriasis. Now, patients who have psoriasis, all of whom show here, have an elevated Th17 response. They produce more of an interleukin called interleukin 23. Unfortunately, there are therapeutics that actually target this pathway, IL-23, for instance, ustekinumab or Stelara. And so we ended up treating patients who have desmogliin deficiency here. Aran Cohen-Barak treated some of his patients with Stelara, and what he was able to see was a dramatic reduction in the skin inflammation in these patients. And we also saw a dramatic reduction in the production of IL-23 within the epidermis. Now, this IL-23 is actually being produced by the keratinocytes. This is not just coming from the immune cells. This is the keratinocytes that are chock full of IL-23 and producing it, and ustekinumab makes that go away. So we were wondering whether or not, if you take this out of the animal or the human and just look in a cell autonomous way in a tissue culture dish, can desmogliin 1 regulate inflammatory cytokines in a keratinocyte autonomous fashion? And to address that, we set up a system in a tissue culture dish whereby we treated cells, keratinocytes, with TNF alpha. And this is known to induce the mRNA expression of IL-23, as shown over here in a dose-dependent fashion. Then we introduced desmogliin 1 into these cells to see what happened to IL-23 expression. We could see that desmogliin expression suppressed IL-23 expression in a keratinocyte autonomous fashion. Now I want to go back and start to link up what I talked about, actin remodeling, with this immune response. And to do that, we're going to use that mutant that I introduced to you, the point mutant that uncouples desmogliin 1 with NIC1, and then presumably that whole actin machinery. What happens to IL-23 expression? Well, lo and behold, it's back up again. So this mutant that uncouples actin from desmogliin 1 is not able to suppress IL-23 mRNA. So how could this possibly be? What is the explanation for this? Well, in 2018, there was a paper that was published that suggested that NWASP, remember NWASP is a critical part of this actin polymerization machinery, also moonlights in the nucleus to inhibit IL-23 expression through an epigenetic mechanism by regulating the suppressive marks methylation on the IL-23 promoter. And so the NWASP knockout mouse that was used in this models in this paper actually had psoriasis. Very interesting. So we're wondering whether or not desmogliin expression through the actin remodeling complex could regulate NWASP. Could it regulate its nuclear transport, its maintenance in the cytoplasm? Basically, could it regulate its distribution? And to address that, we carried out subcellular fractionation. And we saw that in the presence of desmogliin 1, there was ample total NWASP in the nucleus, which theoretically then could suppress IL-23 expression. However, if we knock down desmogliin 1, NWASP in the nucleus goes way down, and what's left is a phosphorylated form here that was known to be able to not suppress IL-23 expression. So this supports the idea that somehow through this actin association, desmoglein 1 can regulate the nuclear to cytoplasmic distribution of N-wasp and thereby regulate IL-23 cytokine expression. So that to summarize here, not only does desmoglein 1 regulate actin remodeling or remodel actin to redistribute tension, which is important for stratification, but it also regulates the nuclear localization of N-wasp to regulate inflammatory cytokine expression. So then just to finish up here, I wanted to go back to just our organ systems in general. So I showed you that in stratified epithelia, desmosomes play an important role in the morphogenesis or multi-layering, but also they actually put a damper on inflammatory cytokine signaling. Now, where in the layers, does this morphogenesis mechanism happen only in the basal layers and perhaps the inflammatory suppression in the superficial layers? Actually, we have some RNA-seq data that suggests maybe that the basal layers are also subject to this inflammation suppression. So going to cardiac and gut, there's also data from a number of laboratories that suggest that inflammation is also an important pathogenic driver in cardiac and gut, and that this could be regulated by desmosomes, not only the Th17 IL-23 pathway, but NF-kappa B, STAT3, and others. And further, it's not just about desmogleins, but also desmoplakin and plakophilins may be involved in these inflammatory pathways. So with that, I'd like to just end up by thanking everyone. And there's so many people to thank, many people who are on this Zoom call today. Thank you. You know who you are, my lab members, other collaborators who are on here. I couldn't really do any of this without you. Thanks go to the current lab members and former lab members of the Green Lab, our cardiac collaborators, our collaborators here at Northwestern and all over the world, and of course, all of our funding agencies. And I'd also like to call out people who I didn't talk about their work today, Bob Harmon, Nat Kroll, and Aaron McCarthy, who work on Dary's disease, and a number of other people. I couldn't really get into their work, but thank you very much. And finally, of course, it's important for all of us to connect internationally, and this ongoing series is something that keeps us connected, and we really appreciate that. Thanks, Claire, again to you and your co-organizers for that. Let's all stick together.